Okay, a little background on gene expression. I'm going to use my double-stranded DNA molecule, the same one I just used. In fact, I didn't even erase it, and all I did was label one side as the gene, and the other side is going to be not the gene. There are DNA nucleotides and there are RNA nucleotides. DNA nucleotides make DNA, this double-stranded molecule that contains the recipes for making the various proteins that the cell or cells need. The DNA nucleotides have 4 nitrogen bases possible: adenine, thymine, cytosine, or guanine. So each nucleotide has one of those bases. It has the sugar deoxyribose and a phosphate group, and that makes up a DNA nucleotide. And there are adenine nucleotides, thymine nucleotides, cytosine nucleotides, and guanine nucleotides. For RNA nucleotides, there's adenine, uracil, cytosine, or guanine, plus a different sugar, ribose, and then a phosphate group. So exactly why there's uracil instead of thymine, I've never looked up. Feel free to do that. But it's obviously a different nucleotide, and it's going to make a different macromolecule, RNA, if it has the sugar ribose. Because that is the difference between adenine, cytosine, and guanine DNA nucleotides, and adenine, cytosine, and guanine RNA nucleotides would be the sugar. That's the only difference between them. And then there's thymine here, uracil here. There are multiple genes along a single double helix. There's a mention of the largest number of, the estimated largest number of genes on a given chromosome, and that was in the text or in my notes. I didn't look that up again. That I'm not going to ask you. Someone then asked me, well, which chromosome has the fewest amount of genes? And while I don't know the answer, it would most likely be one of the smaller ones, like 21, 22, 23, maybe a Y chromosome. Oh yay, my shoulder caught it this time. I'm going to have to come up with another way to do this, because this is just not working as well as I would like it, and I'm going to get hurt when the board falls on me. Okay, so the genes themselves along a given double helix, some are for proteins, and what will happen is the transcript will be a messenger RNA or mRNA transcript that is translated. Some of them are for the other RNAs. So there are ribosomal RNAs that, along with proteins, make up the ribosomes. There are transfer RNAs, and there are about 66 of them. I'll show you that when we look at the genetic code in a second. But there are about 66 of them, and they are made. 2, they are transcribed, as well as some regulatory genes. So it's not one gene per chromosome. There are multiple genes along a chromosome. There are spaces in between that are not what are called coding regions, that are not transcribed, and they may be evolutionary baggage. You can look that up if you're interested in the pieces of DNA that are not transcribed. So anyway, there are multiple ones along each given chromosome, some with more than others. It's going to come off if I do that. Okay. So how does this work? The DNA in a eukaryote is found in the nucleus. So I'm going to give you an analogy of an amazing cookbook. In fact, it's in multiple volumes. For you, it would be, you would have 46 of these volumes. Some of them with slight differences because one came from your mom and one came from your dad, and others with even bigger differences because they have different recipes entirely because they're different chromosomes. But basically, if we can go with this analogy, let's look at one of those volumes. And this is a piece, this is part of one of the recipes that is in that volume. Now, we keep this in the library, which would be the nucleus in my analogy. We keep this in the library so that we do not get it all messy from cooking. Okay, from the actual process of adding the ingredients. That's not exactly why the DNA stays in the nucleus, but this is my analogy. I'm trying to break this down and make it simple. So instead of replication, when the proper enzymes are present and the gene is turned on, yes, it separates again. Okay, so there we go. There's our gene. Over here, is not the gene. And nothing is going to happen with it. Okay? It's there for replication. It's there so that when this is replication, there's a new gene for that second daughter cell. That's what it's for. But this is our recipe. And this is the only one that we're going to transcribe. So basically, with our cookbook analogy, what's going to happen is a note card is going to be made. Instead of moving this volume out of the library, okay, instead of moving the chromosome out and directly translating from the chromosome, the gene side, we're going to just make a note card. But it's going to be like translating it from English to Spanish, except that we're going to go from the DNA language, and this is one that's going to make a protein, to the messenger RNA language. So what happens is, this is an RNA nucleotide. It's an adenine. This one is a uracil. This one is a guanine. Then we have a cytosine, a uracil, an adenine, a uracil, a guanine, and a uracil. This is our messenger RNA. It is not going to stay there. It's going to move away. In fact, that note card, here's a nuclear pore, is going to separate and move out into the cytoplasm. Okay, so that's messenger RNA. Now, it's time to talk about codons. Codons are sets of 3 bases that code for an amino acid. So, I want us to look at the computer for a second. This is the universal genetic code. Okay? That is the universal genetic code that you see there. And these are codons that all these in here are codons. So these are the different sets of 3 that we could see on our messenger RNA. Now, our first one, if you can look up there, is AUG. So A, first place, U, so A, U, G, I don't think third place. So A, U, G, look right there. So I'm matching up the A, the U, and the G that's over here, and I get methionine. That's one of the amino acids. You do not have to memorize that. But that is, the AUG is what's called the start code. So that is the first codon of each messenger RNA transcript. Then we had CUA. CUA, third, is leucine. So LEU stands for leucine. That's an amino acid. Notice that there are multiple codons for leucine. There's AUU. No, isoleucine. Was it what is it? Let me go back to this. CUA. Yes, CUA. There's CUA, CUU, CUG, CUC. All of those codons will put that ingredient in that place in the protein. So, this is how some mutations are silent. Because if it was a switch from CUA to CUC, it'd still be leucine. The protein would not have changed. It'd be like using a different brand of a particular ingredient, but it's still that ingredient. And then finally, we have UGU. So there's U, G, and U, and that one is cysteine, CYS. And there are a couple of codons for cysteine. Notice the stop codons. So let me take this back, and in fact, let me just put this out of the way. Go back over here and go back to initiation, elongation, and termination. There are enzymes involved. Initiation is when in fact, the main enzyme here is RNA polymerase. RNA polymerase binds the gene and starts putting the RNA nucleotides, base pairing them with the DNA nucleotides of the gene. Okay? So when it first starts, the first one, that's initiation. So it's the DNA, the appropriate enzymes for this to get started, and the first RNA nucleotides of the first codon. Then it just keeps going. That's elongation, making the messenger RNA transcript longer. And believe me, none of them are just 9 bases or 3 codons long, but we weren't going to take the time to, do this more realistically. Okay, so elongation. Until on the DNA, there are 3 DNA nucleotides that signal termination. This comes apart. The messenger RNA goes out into the cytoplasm, out of the nucleus if it's a eukaryote. And if there was only one messenger RNA needed, this closes back up. And we're back to this. If not, it keeps making more. So, that's transcription. Transcribing from our nice cookbook volume the recipe that's in DNA language to the recipe in messenger RNA language. Now, again, there are genes that code for ribosomal RNAs and transfer RNAs, but they function in translation. So let me, right up here, transcription. So you know that's what I've been talking about. So primarily right here. Move this without hurting myself, or almost hurting myself again. Okay, this is still our messenger RNA. It's out in the cytoplasm. And what it does is it moves to the countertop where the actual protein, the recipe is going to be put together. So that's what happens. And that countertop is the ribosome. Now the ribosome's in two parts. So let me get another color for my ribosome. So there's one part, there's the other part. Don't worry about the complete accuracy here, but it's the countertop. Okay?